In ancestral gnathostomes or jawed vertebrates, a number of modifications in the spinal cord evolved, which would be retained in amniotes, including humans. The spinal cord was round in cross-section, and the gray matter is divided into the dorsal horns, which process sensory information, and the ventral horns from which motor commands originate. Some of the tracts in the white matter were myelinated. In jawed vertebrates, plexuses form in which the ventral roots of spinal nerves interweave. And from this swapping of fibers in the plexus, the nerves originate, which then innervate the body regions. In gnathostomes, plexuses did form, but they were variable. So, for example, rays have an extensive cervicobrachial plexus, which may involve more than 20 ventral roots. There are more tracts which pass between the spinal cord and brain in jawed vertebrates. The ascending sensory tracts include homologs of the spinotectal, dorsal spinocerebellar, and spinobulbar tracts of higher vertebrates, including humans, and the descending motor tracts include the homolog of the reticulospinal tract. In the spinal cords of osteichthyans, the bony fish, the amount of gray matter is increased. The complexity of the synapses of the spinal cord are increased. The dorsal and ventral roots are closer together, and they always fuse to form a mixed spinal nerve, which is not always the case in more primitive gnathostomes. The soma of motor neurons are located more ventrally. Dorsal and ventral fissures exist in the spinal cord. A definite cervicobrachial plexus exists, and the tracts which are shared among osteichthyans include a new ventral spinocerebellar tract and the descending vestibulospinal tract.